So stem cells, so what it means, stem cell, that means which are going to produce the stem cells, which are going to produce some number of cells, that some types of cells, we can say the stem cells here in general, we define like that. Okay. So in that, first of all, we need to begin with the totipotent stem cell. So that's why I given the general definition. Some number of cells they are going to produce. We call them as stem cell. So totipotent stem cell. What is totipotent? The cells which can produce all types of cells, we can say totipotent cells. Okay, so the totipotent cells. So where do you find totipotent stem cells in our body? So in our body, the question is wrong. We can't find any kind of these kind of totipotent cells here. At least during embryonic development, whatever we said, the epiblast cells which are going to be responsible for the total body. So at the time we can say they are stem cells at the time. Or else if you go little bit back in the zygote, in the zygote. So zygote is responsible for the total formation of the fetus. At least that we can say totipotent stem cells. Totipotent means the cells which can form all types of cells, we can say totipotent cells. Okay. Next, pluripotent. Pluripotent. Potent means capacity. Totipotent, total capacity. Okay, so that pluripotent, that means it was little bit reduced now. If you consider the zygote as all types of cells it formed here, when it comes to the embryonic stages, that epiblast is responsible for only that embryo. Extra embryonic membranes cannot be formed over. So what do you say that as? Look that zygote we said totipotent. So that these particular cells of embryoblast we can say pluripotent. That means limited to some number of cells. Okay. Next, multipotent cells. The limitation is much increased. We can say multipotent. Only a certain type of cells are going to be produced over. We can say multipotent. Okay. Next, unipotent stem cells. Only one type of cells are going to be formed over. Hemopoietic stem cells, which are going to form all the blood cells, is the best example for multipotent stem cells. Unipotent, you may learn over in your earlier classes. In muscular tissue, we said satellite cells are responsible for the formation of skeletal muscle fiber. Pericytes are the regenerative cells of the smooth muscle fiber. That means pericytes meant for only smooth muscle fiber. What do you say them as? Unipotent stem cells. Okay. Finally, non-potent stem cells. Non-potent. What it means? Non-potent stem cells means the cell which cannot form any type of cell. We can say the cell which cannot they're able to produce any other type of cell. We call non-potent stem cell. There are so many cells in our body to say they are non-potent stem cells. Nerve cell. Non-potent, you can't say stem actually, stem cell. Okay, so we categorize in the form of totipotent, which will be seen over an only early embryonic stages, or else you can take zygote. Then after the capacity embryo development is going on, that cells will be restricted to a certain type, pluripotent. When development is going on, that will turn to multipotent. When development is going on, then only unipotent stem cells we can see. Then finally, that most of our body is with non-potent stem cells. Okay. So this is a basic classification of potency of formation of cells here. So if you observe, these kind of totipotency you'll find in case of sponges very high. That means in lower invertebrates, if you cut the sponge into the many pieces, each piece is able to develop the full sponge again. Why? Because the totipotent cells are present in the adult also. If you do with the same with human being, is it possible here if we cut the human being into pieces, is it possible here that is able to produce the full human body, that a piece will able to produce full human body? No, not at all possible, isn't it? So that what you can say here, that the human being lacks these kind of totipotent cells. You will find this type of totipotent cells in case of earlier invertebrates. Even the evolutionary process is going on, adult stage is able to produce everything is lost. Okay, so why we are discussing this? That is to discuss about the hemopoietic stem cells. So our topic is how the blood cells are going to be formed. So that is our topic, hemopoietic stem cells. That we need to discuss about the hemopoietic stem cells. That in general, hemopoietic stem cells are considered as that multipotent cells. Multipotent cells, we can say hemopoietin cell, because they are going to form RBC, WBC, all types of blood cells, we can say simply. Okay. So that is the story of hemopoietic stem cells. We have a beautiful chart in the text. We'll shift to the chart and then we'll discuss the remaining issues there. Okay. So here it is. Hemopoietic stem cell. Hemopoietic stem cell, which is considered as actually multipotent stem cell, which is given in the theory, but in a diagram they said it as pluripotent stem cell. Just keep that in mind. What is fact means hemopoietic stem cell is multipotent stem cell. Okay. What is multipotency? What is pluripotency? That we already said over. Zygote is a totipotent. We can say totipotent. Next level pluripotent, then multipotent, then unipotent, and finally most of the cells are without any potency. Okay, so here it is a hemopoietic stem cell. Okay, so what is hemopoietic stem cell? That's going to be the question here, that we need to correlate with our the bone information we learned in first year. The bone is in the spongy bone, we say there is a red bone marrow. The spongy bone is filled with red bone marrow. Between the irregularly dense arranged matrix, that red bone marrow is present. So that red bone marrow is having these kind of stem cells. Okay, so these kind of stem cells. What do you call the stem cells? As hemopoietic stem cells. They are called as hemopoietic stem cells. Okay. So these hemopoietic stem cell is differentiated into during the formation of blood cells. So who are the blood cells? I don't think so. I have to remind you. RBC. And in the WBC, granulocyte, agranulocyte. In the granulocyte, eosinophil, basophil, and neutrophils. Agranulocytes are of lymphocyte and monocyte. All these blood cells are going to be formed from the red bone marrow. In the red bone marrow, particularly, we are talking about hemopoietic stem cell. So, for the formation of all the blood cells, hemopoietic stem cell differentiated into myeloid stem cell and lymphoid stem cell. Myeloid stem cell and lymphoid stem cell. Okay. So let's take from here. So myeloid stem cell is going to form that RBC, platelets, basophil, eosinophil, neutrophil, monocyte, the dendritic cells. All these are formed from myeloid stem cell. It's difficult to remember, isn't it? So you just remember the lymphoid stem cell, because lymphoid stem cells are going to form lymphocytes. Okay. The remaining, what remains? RBC and all granulocytes like acidophil, basophil, neutrophil, and monocytes. Agranulocyte, no monocyte. And next, RBC, everything formed from myeloid stem cell. Lymphoid stem cell form lymphocytes only. Before we enter inside, we need to know this word, self-renewal. What is self-renewal? For example, if you find these kind of 100 hemopoietic stem cells, these 100 hemopoietic stem cells are divided in the mitosis and form some 200 cells, isn't it? In that, the mitotic division, a cell divides and form 200 cell divide means 200 cells are formed. So in that, 100 cells again act as hemopoietic stem cell, and the remaining 100 cells only form the myeloid and lymphoid stem cell. That means after the formation of all the blood cells, even you will find 100 hemopoietic stem cell. There is no reducing number here. So that what do you call self-renewal cells. Hemopoietic stem cells are self-renewal. It doesn't happen with myeloid stem cells. So what do you call them as non-self-renewal? Okay, so that means in case if you in the red bone marrow, if 100 stem cells are present, after the formation of myeloid and lymphoid, also you'll find the same 100. But in a period of time, while age is growing, that the power of these particular stem cells, what power, division power, is going to be reduced over. That's why in old age we used to say that blood cells, the count problem will come over. Okay. So then that is self-renewal. So what I can say, hemopoietic stem cell is a multipotent stem cell and it's a self-renewal stem cell. And it is given here in the chart as pluripotent. That also need to be remembered over. But the theory is given correct, multipotent. Okay. Next, myeloid stem cell. We can say these are myeloid stem cell. What they do? What they do? They form the progenitors. Erythrocyte progenitor, megakaryocyte cell progenitor, basophil progenitor, eosinophil progenitor, granulocyte monocyte progenitor. That erythrocyte progenitor form RBC. Megakaryocyte cells are going to blast out and form the platelet because they are fragments of megakaryocytes. So basophil committed progenitor form the basophil. You know that areolar connective tissue containing mast cell also nothing but basophil. Next, eosinophil committed progenitor form eosinophil. Granulocyte monocyte committed progenitor is committed to form neutrophils and monocytes. Okay. Next, so without formation of any progenitor, you can see here myeloid stem cell form directly dendritic cell. Dendritic cells are considered as antigen presenting cell. No way they are related to nervous tissue. They look like neuron with having dendrite-like branches. That's why they call dendritic cell. But they are antigen presenting cells that we discussed in immunology. Okay, so dendritic cells we can say. Okay, next. They are directly formed, no progenitor. Both myeloid and lymphoid stem cells are directly formed. Okay. In the same way, the lymphoid stem cells form the B cell committed progenitor and T cell committed progenitor. The B cell committed progenitor form B cells, T cell committed progenitor form T cells. That T cells are of two types, T cytotoxic cells and T helper cells. T cytotoxic and T helper cell. Okay. Then here itself, we need to discuss about the some sequences here. If we want to form the sequence of RBC, will come like this. Here it is the hemopoietic stem cell. Hemopoietic stem cell form myeloid stem cell. Okay. Myeloid stem cell form this one. Erythrocyte progenitor. I should write the total. Okay. Erythrocyte progenitor. This erythrocyte progenitor form pro-erythroblast cell. This erythrocyte progenitor form pro-erythroblast cell. Pro-erythroblast cell develops into erythroblast cell. Erythroblast cell develops into immature RBC we call reticulocyte. Reticulocyte form the RBC. This is a full sequence actually. They are little given here. But the formation like this. Hemopoietic stem cell form myeloid stem cell. From their erythrocyte progenitor is formed. Erythrocyte progenitor developed into proerythroblast. Then it develops into erythroblast. Then reticulocyte. Reticulocyte is having all cytoorganelles actually. That it loses all the cytoorganelle and that develops and mature into a red blood cell. This is the story of the erythrocyte. In the same way, when you want to talk about the platelets, how the platelets are formed. Hemopoietic stem cell develop into myeloid stem cell and myeloid stem cell form the megakaryocyte cell and that megakaryocyte cell is going to break down and form the platelet. Okay. Next, the basophils. How does the basophil and mast cells are formed? That hemopoietic stem cell, let me write here, hemopoietic stem cell form myeloid stem cell, then that form the basophil progenitor form basophil. Okay, so I think you understand what I'm saying. This is for eosinophils, and this is for that hemopoietic stem cell, myeloid stem cell, and the granulocyte monocyte progenitor form the granulocyte called neutrophil, and also it formed the monocyte. That monocyte become macrophages, that we know the story of macrophages. Okay, next, directly dendritic cells are formed. Both hemopoietic, both myeloid stem cell and lymphoid stem cell, both are formed this dendritic cell. Okay, so this is the story. And here, the B cell progenitor form B cell, T cell progenitor form T cell, that we said, isn't it? So that we give the conclusion for that. That will give the conclusion for that. We have basically that there are three steps here in the formation of in the activity of these immune cells. They are immune cells. That's why we need to take a little bit here. Production, maturation, and activation. Both B cell and T cells having three major steps: production, maturation, activation. So about T cells. T cells are of four types, but in our text it was mentioned two types: T helper and T cytotoxic. That we'll discuss in immunology. Okay, so this is a table. Okay, in this table, we need to see. Uh, Let us maintain little decency in not using scale, but I hope may get much better. Yeah. So production. So production of T cells and B cells both happened in the red bone marrow. We may write here. Okay, red bone marrow. In the red bone marrow here also. The red bone marrow. Exactly where in the red bone marrow that we are seeing now. That exactly they are formed from hemopoietic stem cell which form the lymphoid stem cell. Hemopoietic stem cell which differentiated into lymphoid cells. Okay, that is called production. So which is happened in the red bone marrow. After that, T cells are going to shift into thymus. The T cells are going to shift into thymus. So maturation of T cells occur in the thymus. Maturation of B cells occur in the bone marrow. Okay, so by the maturation only, we are given the names like T cell, that means thymic lymphocyte, B cell, that means the bone marrow lymphocyte. That means the meaning of B is bone marrow, meaning of T is thymic lymphocyte. Okay, so thymic, what means? Based on the maturation, we are given the name. Activation. Activation occur in the secondary lymphoid organs. Activation occur in secondary lymphoid organs and secondary lymphoid tissue. Activation require microbes. When microbes enter, that means these are trained army, you need to understand up to here. So then after they'll go to the different places, secondary lymphoid organs and secondary lymphoid tissues. Well, they go to that particular places and there they do the activation. Lymphoid organs and tissues are said to be that spleen. We can take example. We can take example of spleen. So let me write here. Need to increase little font size. 36 is enough. Okay, spleen. You can say lymph nodes. Spleen, lymph nodes. They are all said to be secondary lymphoid lymph nodes. Okay. Next, MALT, mucosa associated lymphoid tissue. We can say, we'll discuss that back in immunology, mucosa associated lymphoid tissue. That means the mucosa layer, wherever it present, these cells are going to be settled down there and they do the activation there. Killing of microbes we call activation. So in the table format, if we draw that, we'll understand much better. In that way, that about the stem cells, they are shown here. So this is all about the story of stem cells. Okay, so this is the story of stem cells. So here, this area I get much elaborated, and I just made a sequence of red blood cells. This area only is shown in the form of a table. Okay. Then the sequence for erythrocyte that to be remembered over hemopoietic stem cell, myeloid stem cell. From there, that erythrocyte progenitor that form the proerythroblast, that form the erythroblast, that form the reticulocyte. Finally, erythrocyte or RBC are going to be formed. Okay, what is the importance of the stem cells here? Over a period of time, when age is growing, the vigor and vitality of the cells is going to be reduced over. You see, these are all immune cells. So that the immune cell production also going to be reduced over in old age. Okay, thymus, if we come over that earlier it disappeared. Thymus is a child gland where T lymphocytes are going to be matured over. So over the time age is growing, the vigor and vitality of this particular hemopoietic stem cell reduced. That's why quality of the cells also going to be reduced over. Nowadays we got the technology that stem cell replacement, stem cell technology. Imagine for an old age person, if we are given that new stem cell. So what he is able to do, he is able to produce all these cells with great vigor and vitality, isn't it? So that means RBC, that whatever their functions, you know them, that how they participate in the vital activities of the body. So automatically, vital activities of the body is also going to be increased over. Okay. So few of the stem cells in our body, let us remind, not only red bone marrow, few of the stem cells. That means skin. If we take, we learned in first year that stratified squamous keratinoid is having above the basement membrane, there is a germinal epithelium. We call that is stratum germinatum, which is going to produce the cells continuously. That the cells are polyhedral cells. When they go up, they become squamous. The cells will become squamous and they die off when they go out. So what do you call it as stratified squamous? That is our skin, that is our hair, our nails. All these are belongs to the stratified squamous keratinoid. What happens when the age is growing? These germinal epithelial stem cells are going to lose the vigor. That's why they are not able to produce the new cells. So that's why the folds on the skin. So what is the solution then? The stem cell technology is going to replace them. That means at the 100 plus also, if you make the technological advancement, if it happen in future, 100 plus also, you look so young like 25. That means that is the importance of the stem cell technology. That advancement is going to happen. Just it's a now at present it's a theory. It may happen in future. All these things. Okay. So that's about the stem cells. That uh, if you have any doubts here, you can ask me.